this video, we will be going through lipids, which is a part of the biological molecules topic. There are two types of lipids that you need to know about, triglycerides and phospholipids. It is important that you are able to compare the two structures and functions for your exams. Triglycerides are made up of the monomers glycerol and fatty acids. These fatty acid chains can either be saturated or unsaturated. The only difference is that unsaturated fatty acids contain carbon-carbon double bonds within its chain. These are found within plants and are mainly seen in the form of vegetable oils. They are liquid at room temperature because the double bonds cause kinks in the chains, which disrupts the intermolecular bonds that can form. Saturated fatty acids have single carbon-carbon bonds and are mainly seen as solids in the form of animal fat. The bonds which form in lipids are called ester bonds. They are joined by a condensation reaction where two monomers come together and a water molecule is removed, leaving a covalent bond. Although this is a condensation reaction, with lipids it is termed esterification. If you wanted to reverse this and break the covalent bond, this would be the hydrolysis reaction, where you need to add a water molecule. Triglycerides have many functions. They are used as energy storage and are more efficient at this than carbohydrates. As they are hydrophobic, they do not cause any osmotic shifts in the cells. They are a source of metabolic water as the oxidation of the carbon-hydrogen bonds releases water, and camels, for example, use this in the desert where water is hard to come by. Triglycerides provide insulation in the form of myelin sheaths around neurons, as well as part of adipose tissue, preventing heat loss too. Finally, triglycerides increase animal buoyancy. Due to their low density, it enables animals to float easily. Now, phospholipids. These are also formed from the monomer glycerol and fatty acids, but instead of having 3 fatty acids like triglycerides, they only have 2. One of the fatty acids have been replaced by a phosphate ion. Now, as the phosphate is polar, it makes it soluble in water, making it hydrophilic, but the fatty acid tails are still hydrophobic. Due to this adaptation, phospholipids are the main component of cell surface membranes as they form a phospholipid bilayer with a hydrophobic fatty acid core. Again, as with triglycerides, the fatty acid chains can be saturated or unsaturated, but the presence of an unsaturated chain will make the membranes more fluid-like due to the weaker intermolecular bonding. Finally, in order to test for the presence of lipids, the emulsion test is done. This is where alcohol is added to the sample and shaken. If the new solution looks like a milky white emulsion, then lipids are present. Even though lipids are hydrophobic, they will dissolve in alcohol. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.